Hi, my name is Shouri, and my SURF 2023 research project was on synthesis of a fluorescent marker for imaging senescence in specific cell populations. All right, so first, what exactly is senescence? Senescence is a state of the cell in which the cell is no longer dividing or proliferating. Senescence is implicated in various disorders like cancer. It can be a defense mechanism against cancer cells to prevent them from proliferating, or it can be something that the cancerous cells have to overcome in order to proliferate and grow. It's also, of course, used to prevent the growth of cells that have accumulated a large number of mutations, again, along with the same theme as cancer. It's also implicated in various aging-related disorders, as in these disorders, certain cells may not function properly within the tissues because they have stopped dividing and providing new cells to the tissues that have been aged. And so as a result, many researchers are interested in quantifying and detecting senescence within human and animal cell lines. One such molecule or enzyme of interest is SA-beta-gal. Researchers have found that senescent cells tended to have a much higher concentration of SA-beta-gal in comparison to actively dividing cells. My research focuses on detecting SA-beta-gal and thereby senescence within specific cell populations. The specificity of the cell population is ensured through a peptide side chain. And the peptide side chain is selected such that only certain cell populations can cleave it. So the mechanism for this molecular probe that was synthesized in my research project was first the target enzyme within a cell needs to cleave off the peptide side chain. After that, SA-beta-gal needs to cleave the beta bond at the C1 position. This leads to a resonance structure, and finally, an intracellular protein binds and allows the fluorescent dye / probe that was released from the initial molecule to be retained within the cell. This allows for fluorescence to be turned on within the cell for a greater period of time, allowing for greater longevity when imaging senescent cell populations. All right, so now onto the synthetic route. This was the synthesis to produce the molecular probe or large fluorescent dye. And so, as you can see, most steps involve modifications to the groups on the C1 position. The first step of the reaction adds protecting groups through to carbon C1 through C4 as well as C6. C6 has a tritial group because this facilitates for an easier attachment of the peptide side chain. After all the protecting groups are added, the acetyl group on the C1 carbon is converted to a hydroxyl group. After that, CBr4 is added to, and this allows for bromine to be added in the alpha position. It's very important to add CBr4 instead of HBr because this facilitates for the bromine group to be added in the alpha position. And because the bromine group is added in the alpha position through a glycosylation reaction, a benzaldehyde group, hydroxybenzaldehyde group can be attached in the beta position of the C1 carbon. And it's very important that our final fluorescent molecule that is to be released within the cell is added in the beta position because this allows SA-beta-gal to actually cleave that molecule and once again release it into the cell. After the hydroxybenzaldehyde is attached, a reducing agent, NABH4, is used to reduce the aldehyde to an alcohol group. Then afterwards, a bromination reaction takes place, and finally, the the fluorescent marker that is meant to be released from a larger molecule is created from IR780, and that molecule is added via a nucleophilic attack in addition. And most of the yields for the reactions were fairly high, were around 60 to 70%. There were two reactions that had a slightly lower yield. This one, the retroaldol condensation of IR780 had only a 21% yield, and the addition of NaOH and hydroxybenzaldehyde had a 35% yield. So those are two reactions that may need to be optimized further, or other routes may need to be explored in order to get around the low yields in those reactions. All right, so now on to my methodologies. For each of the reactions present in the reaction scheme, they followed the general process of first, of course, the reaction, monitoring the reaction using TLC to determine that the product had been formed. Afterwards, isolation using column extraction, and then isolation using column chromatography, and finally characterization using mass spectroscopy. And mass spectroscopy is one of the instruments within the LCMS machine. For the column chromatography, silica gel was used, and the stationary phase and the mobile phase consisted of an organic solvent. The gradient of the organic solvent mixture was slowly increased to be able to elute out specific fractions, and of those fractions, the fraction that contained the molecule of interest was obtained using analysis with thin layer chromatography. And then afterwards, characterization was performed using the mass spectroscopy to determine that once again confirm that our product of interest was isolated, mainly to ensure that there were no other major side products that were generated within our fraction. Finally, I would like to thank Laura for being my mentor, as well as Dr. Coy for hosting me in her lab for the summer of 2023. I would also like to thank the SURF program for hosting the summer research session, as well as all of the administrators and faculty that made it possible. Thank you so much for attending my presentation.